So next up is Kevin Jennings, and Kevin's with the New York State DEC. He's going to talk about some changes in the New York State freshwater wetlands regs that affect vernal pools. Hey, thanks for coming.
All right. Do I hit that? There we go. Gotcha. All right, very good. All right. Thanks for having me, Mike. So my name's Kevin Jennings. I am with the Division of Fish and Wildlife here on Stony Brook campus with the Region 1 DEC office. Don't be worried about the title. This is not just about my career here at DEC. Not that important, but we are going to talk about some unusually important wetlands and the expanded freshwater wetlands protection that came with the change in the New York State Freshwater Wetlands Act. Just a little more than a year ago, January 1st, 2025, the new freshwater wetlands law went into effect, and so we got rid of the regulatory maps. One of the problems with regulating freshwater wetlands is that, wetlands were protected if they were greater than 12.4 acres in New York State, but they had to be on the map. And so part of the problem with making maps is that they're not always right, and they were very difficult to amend. So what happened was there were lots of missing wetlands, and so for a long time, DEC wanted to get rid of those maps and provide a different way for protecting wetlands. The criteria for protecting wetlands, right, like I said, is it's over 12.4 acres right now. In 2028, that will move down to 7.4 acres, so we will be protecting even more wetlands come 2028. But there's also 11 criteria that are explicitly laid out in the law, known as unusual importance criteria. We have been able to protect wetlands of less than 12.4 acres over the years. It used to be called ULI, unusual local importance, and that was changed to now just be unusual importance. It was pretty generic in the past. It was pretty broad for which wetlands can be picked up as ULI. On Long Island, there was a big effort led by Steve Sanford to protect a lot of these small wetlands on Long Island. So we do have a lot of wetlands that are already mapped in the past on Long Island, less than 12.4 acres as ULI wetlands. But the new change in law really explicitly laid that out, and we'll go through some of them. So what does unusual importance mean? What does it mean? It is just, it shall mean a freshwater wetland regardless of size, right? So it doesn't have to be any particular size, that possesses one or more of the following characteristics, and that's what we're going to look at today. We're going to go through all the 11 characteristics, but we're going to try to focus on some more particular ones, particularly the productive vernal pools one. So there are two of them that are pretty straightforward, right? So previously designated as ULI, if it was already designated as an unusual local important wetlands, it's going to be regulated now, so it's pretty straightforward. Previously mapped, if it was on a map, again, pretty straightforward. No changes there. They're still going to be regulated. Then we have a handful of others that were laid out in the statute that needed some clarification. What does it mean to be one of these things? And so that was determined, put out in the regulations, and we'll talk about these a little bit. And like I said, the one I really want to talk about today and focus a little more time on is productive vernal pools and how they're being protected. So, is it there? Oh, okay. I didn't know it was this. Going to be one at a time. So what does it mean to be a productive vernal pool, right? So I'm just going to probably put them all up here so everyone can see them. So a productive vernal pool, it has to be a productive vernal pool, right? So what does it mean to be productive? Harrison mentioned the main criteria, but New York State has their own criteria. So it is based on some work that New York Natural Heritage did. And there's different criteria based on egg masses and or larvae of particular species. Right now, there is no list, and the process for getting vernal pools protected will be, it's something the DEC is working on, and there'll be a process for individuals to get productive vernal pools on this list. But it needs to be a productive vernal pool, it needs to be known by the department, and the department will be listing those on the DEC's website and publishing them in the environmental notice bulletin. So what does it mean to be productive? So first we're starting with some of the common species that you find in vernal pools. We got spotted salamanders and wood frogs, And on Long Island, it really is just, we're just looking at egg masses. So if you have these other areas, right? So, nope, that's not what I wanted. Yeah, other areas, which consists of Long Island, right? As long as you have more than 10 or more spotted salamander egg masses, it's considered a productive vernal pool. And if you have more than 15 egg masses for wood frogs, it would be considered a productive vernal pool. Looking at some of our special concern species, the Jefferson, we don't have Jefferson salamander on Long Island, but blue spotted salamanders, as long as you have one egg mass or larvae documented in the vernal pool, it would be eligible for protection as a productive vernal pool. Furthermore, we have tiger salamanders, which are currently listed as endangered in New York State. Marble salamanders are also designated special concern, but either of those, if you have anywhere that they're found in the state, obviously tiger salamanders are only right now found on Long Island, but anywhere in the state, as long as you have one egg mass or find one larvae, these would be considered protected. So, like I said, the process is still being worked out on how we go about documenting these, but we will. But part of documenting these is going to be, does this overlap with other criteria? It doesn't need to necessarily be a productive vernal pool if it lands in one of those other criteria. So we'll talk about some of those other criteria, and it might be helpful. We talked about this a little bit in the vernal pool working group, but being able to hone in some efforts on trying to figure out where the productive vernal pools are in areas where they are not protected by some other unusual importance criteria. Okay, so tiger salamanders, right, like I said, make up one of the productive vernal pool categories. So what we've been doing, we've been looking at tiger salamander presence and absence at sites on Long Island for probably since the 80s. But some of those sites haven't been documented or been searched for a number of years. So sometimes it can be very difficult to get to these sites, and we just don't have very much information. So originally, we didn't really think that, we do the traditional surveys, egg mass counts, nighttime counts for tiger salamander adults, doing staining for larvae. But so we know how to find tiger salamanders and document their presence and absence, so we weren't so sure that this would be something useful. However, we don't always have staff. It's not always good timing to get out there and look at these ponds. And we're trying to figure out whether or not using eDNA would give us the ability to be more efficient in documenting presence absence of tiger salamanders at these ponds. And not just that, but if we know where they're at, and we might be able to say something about trends. So if you know anything about the New York State Wildlife Action Plan, in order to get federal money, we need to have a state wildlife action plan, and every 10 years, and try to assess what's happening with the population in New York. And being able to do surveys at these sites will help identify those trends. However, we just don't really get out that often to be able to say anything really concrete about what's happening at these ponds. Not just productivity, but also how the population is doing at those sites. So we had the opportunity to work with folks at Cornell Wildlife Health, and it's a little different than Harrison's talking about doing metabarcoding. We were doing the qPCR, so it was very specific technique for identifying tiger salamander DNA in these pools. That was the first year, just trying to figure out, Cornell was working on getting that qPCR technique down. Then we had a field season of trying to figure out what is the most appropriate volume of water to collect, what's the most appropriate filter to use, and then also, and then once we narrowed that down, then we tried to figure out, well, how many times do we need to go to these sites, right, in order to feel confident about our results. When we get positive DNA results at the site, we know that they're there. But if we get a negative, how confident are we that that's actually a negative result and not just, we were there at the wrong time? DNA does degrade, and it may not be there all the time. We don't have a good sense of how long it's there or how much it degrades at these sites. But so we wanted to get a sense of how often do we need to go. And so let me just bring these up. So we wanted to look to see whether or not these can, this technique can, right, help us identify productive vernal pools. Again, not just for not just for the sake of their productivity, because you can't really tell much about the productivity from doing, collecting water samples and identifying whether or not there's tiger salamander DNA there. You can get a sense of the quantity of DNA that you collect, but we don't really have a good sense of what the productivity is. But it could help us focus more intensive surveys using traditional techniques at those sites if we can sort of hone in on which sites are where they're present. And then being able to go out and know that they're there on any given year might help us establish trends. Like I said, having staff, not having staff every year at right time of the year to go out and do it, it's hard to be consistent and really understand what's going on at a site in any given year. So being able to do this, and on top of that, going out to the sites, we often are sending, not just, with taking water samples, you could send one person. Most of the time we're sending staff out to do surveys. There are spots that are not really easy to survey. They're pretty mucky, they're deep. Sending staff out one at a time isn't just generally good practice. So if we don't have staff where we can do full-on traditional surveys, it is helpful to be able to get some information where we can send one person out, collect water samples, filter them, and have Cornell run some testing for the tiger salamander DNA. And so what we found was, after looking at sites, we looked at a bunch of different sites based on density throughout the year, this past year, trying to figure out when we did them throughout the course of the year from, around February when we find them entering the pools to start breeding, all the way through the end as the larvae leave the pond, so as adults. And what we found was that in general, if they're there, we're finding the DNA early in the season. But sometimes there can be a miss right early on. So we were then finding them during the spike when they're there as larvae, right, later in the spring. So what we recommend is probably what we are going to do in the future is go to the sites early in the late winter as the adults are entering the pools, and then if we don't get a result, going later in the spring as larvae are there and presumably shedding more DNA. And so the idea, again, would be to sort of be able to get a little bit more information about trends at these sites, whether or not they're there, are they still there, how often they're there in any given year, and then maybe they focus more intensive surveys at those sites because of that. Okay. So one of the other criteria, I'm just going to kind of now go through some of the other criteria that establish what makes it unusually a wetland of unusual importance. So urban area is one of them, right? So Long Island, not just Region 1, Nassau and Suffolk County, but New York City I included in here as part of just this quick analysis. The urban area is in blue, and there are, so the significant portion of the island is urban area, and you find pockets in different areas, also a large section on the Pine Barrens, and then some of the ag stuff on the North Fork, and then also some other areas out in the South Fork. So this is what I was talking about, trying to hone in where we're going to do surveys for, are there productive vernal pools? Well, if it's in any of these urban areas, it's not necessary, in order to protect those pools, it's not necessary to document their productivity because they're already going to be, as long as they have wetland vegetation, they're already going to be protected because they're in an urban area. So you can see the sort of the relative, to the rest of the state, downstate, Long Island, New York City is pretty heavily dominated as urban area. So 79% of the island, both Region 1 and 2, urban area, 74% of Region 1 is urban, and nearly all of Region 2, New York City, is an urban area. Significant flooding, so wetland is unusually important, obviously in areas where there is significant flooding, so any wetland is helpful. So how do you determine that? the department decided that in order to figure it out, we need to look at, like, well, where are the areas where there's a lot of impervious surface, where there's going to be a lot of runoff, where there's not much water storage in an area, and areas where there are people, right? Because flooding is going to affect people and resources. So if those three things come together, those are going to be those watersheds that are going to be important for unusual local importance. As long as so you see like the impervious surface, again, not all these areas overlap, but any of these watersheds that overlap with these three categories would give you the result down at the bottom. So I just sort of zoomed in on Long Island here, and these purple watersheds are the ones that overlap with all three of these criteria being met. And then an area that shows up in a floodway. This is just, it's straight from the FEMA's flood insurance rate map. Not too many floodways on Long Island. What happened there? Unusual importance, again, like if there's rare plants or rare animals. So rare plants or anything that would, if you're familiar with the natural heritage nature serve model, we're talking about sites that are, plants that would be considered S3. So they're less than 35 sites. And less than 5,000 individuals. And also, if there are rare animals that use that wetland, so any endangered or threatened species, so automatically tiger salamanders, because they're endangered if they're using a wetland, it's going to be an unusual importance wetland. And then anything that's sort of found in our species of greatest conservation need, found in our state wildlife action plan. If they are wetland dependent species and habitat loss was considered a moderate or heavy threat or high threat, it would be considered a unusual local importance. Unusual importance. Haven't quite made the change yet. And then some just additional class 1 characteristics, whatever the other class 1 characteristics are. If it has a class 1 characteristic, it would be unusual importance. There are 9 in total. Some of them overlap with the other UI criteria. Being connected to tidal wetland is one of them, nutrient poor wetlands. So if it's class 1, meaning it needs the highest protection, it would be considered unusual importance regardless of size. And just the last two, there's some sort of catch-alls for being able to get some things that are, to make sure that there are any other wetlands out there. If they haven't met all those 9 other criteria, there's still these 2 last criteria you can get caught up in. One being local significance, and local significance was just defined as has wetland functions and values that are of local or regional significance. What does that mean in practical terms? Well, the DEC designates, or towns or other municipalities can designate critical environmental areas for a number of different reasons. If it's been designated as a critical environmental area and there's a wetland there, a wetland of any size would be regulated. And then as far as protecting the water quality of the state, the commissioner has the ability to sort of determine, that there's these wetlands, regardless of size again, so small ones that are significant for the importance of protecting the state's water quality. And that just means, this is related to the water quality standards and guidance values. So, things like, total maximum daily loads for different sites, making sure that in order to meet these water quality standards, this wetland is important for making sure that these water quality standards within these water bodies are met. So that can be made by the findings by the commissioner. So just, quick wrap up, just 11 unusual importance criteria. It only needs to meet one of them in order to be designated as a UI wetland. And if any one of these are met, less than 12.4 acres in 2028, it'll be less than 7.4 acres, then a wetland would be regulated, again, regardless of size. Don't have much time for questions, but I am done, so if you think there's questions, we probably gotta get back on time, so thank you.
Or is just the water quality usually too, Have you considered using drones to help survey the ponds that look for egg masses? Like you said, some of the areas are really muddy or really deep.
Yeah, no, we have not considered drones. It's an interesting idea. I'm sure there's some practical difficulties with flying drones in sort of forested areas, but and being able to see well in waters that are sometimes turbid, sometimes really dark, just due to tannins. With the egg masses being, kind of below the surface, it might be difficult with glare and whatnot to see them. But it is an interesting idea to figure out other ways to survey with limited resources. Yeah.
Thank you, Kevin.